This protocol allows us to answer a variety of questions about the way that shear acts on cells in solution and provides significant control over shear duration and magnitude. Unlike other techniques, which detect the effect of shear on cells that are immobilized, this technique allows us to apply shear to cells in solution and lends itself to a spectrum of downstream signal detection methods.
After obtaining peripheral blood from a consenting healthy adult donor in a 4.5 milliliter tube of 3.8% trisodium citrate, centrifuge the blood to obtain platelet-rich plasma and use a pipette tip cut at a 45-degree angle to carefully remove the resulting top, cloudy, yellow platelet-rich plasma layer. Obtain the platelet count via a complete blood count and adjust the platelet count to approximately 2.5 times 10 to the 5th platelets per microliter with pooled human platelet pore plasma. Then place the suspension at 22 degrees Celsius with gentle rotation. For ligand treatment, slowly add the antibody or ligand of interest to the platelet-rich plasma and mix the platelets gently. During the incubation, turn on the cone plate viscometer and set the plate temperature to 22 degrees Celsius. After 5 to 10 minutes, transfer the treated platelet-rich plasma onto the temperature-controlled cone plate viscometer directly at the center of the plate, taking care that all of the sample is deposited between the cone and plate at the point of contact. To apply shear to the sample, calculate the shear according to the viscometer manual, and apply shear to the sample at the appropriate rate and duration. At the end of the application,